Hi guys. So I see that maybe even today we will work really in a very small number. Nevertheless, yes. Why not? Do you have any questions, guys, before we start?
No question from me.
Okay. Uh, Perfect then, guys. Usual structure. Firstly, introduction, some introduction quiz questions to recap what we already learned, and then we will move further where we just ended yesterday. Um, yes, and then we will continue today with the classical Mendelian genetics, and it will be also a part of today's core. There are three Mendelian laws, and we will have a deeper look on them. Okay, so if you do not have any questions, let me shortly start. And I would like to ask Tomohiro, can you read?
Which enzyme is primarily responsible for the process of transcription? A. DNA polymerase. B. RNA polymerase. C. RNA helicase. D. DNA helicase.
Okay, let's do have a poll. Okay, so here are the results, so let's discuss them. Tomohiro, what have you chosen?
I chose A.
Okay, and why?
Uh, I remember that polymerase enzyme is responsible for transcription, but which polymerase is responsible is I don't remember, so I chose A.
Mm-hmm. Okay, you chose A. Hitomi, what about you?
I chose B. Um, RNA polymerase tra- transcribe DNA to mRNA.
Mm-hmm.
So I chose B.
Okay, let me show you the picture. So if we get back to the transcription and we see this scheme, this picture on the slide, and we zoom in a little, we see that for transcription, RNA polymerase is responsible for. Okay, why RNA polymerase? RNA polymerase is enzyme primarily responsible for the process. During transcription, RNA polymerase binds to specific DNA sequence as we see here uh, as the promoter, which marks the starting point for transcription. It then unwinds unwinds the DNA double helix. Okay, so basically unwinding means it's breaking down the bonds between two DNA strands and uses uh, and uses one of the DNA strands as a template to synthesize a complementary RNA strand, as we see here. Okay, mRNA is an outcome of RNA polymerase activity. Um, yes, and then mRNA serves as a template for protein synthesis in the translation. So if we get back to our question, I have to zoom in a little. DNA polymerase, RNA polymerase is correct. DNA polymerase is involved in DNA replication, not transcription. Okay, so in DNA replication, if we zoom in a little again, we see the DNA polymerase is involved during the transcription and uh, then synthesizing complementary new DNA strand on leading as well as on lagging strand. The other options, the other options like RNA helicase is so far not known enzyme involved in transcription. Helicases in general are responsible for our unwinding the DNA double helix during various cellular processes. So basically, if you see some mm, enzyme which is named helicase, it will very probably do the unwinding or breaking the bonds between the DNA strands and basically unwinding the double helix structure. Okay, so RNA helicase is not known so far that it is involved in transcription. However, DNA helicase is also involved in DNA replication, and it does um, uh, and it does the unwinding process. Okay, so mhm. If we get back one more time, here on the first picture, we see helicase or DNA helicase, how it is involved and what is basically doing with the DNA double strand. Okay, it's do this unwinding. Okay, so that was the first question. Let's go further. Uh, Hitomi, please, can you read?
Which enzyme is responsible for unwinding the DNA double helix during the process of DNA replication? A. RNA polymerase. B. DNA ligase. C. helicase. D. DNA polymerase.
One more person? Okay, so here are the results. So let's discuss them. Hitomi, what have you chosen?
Um, I chose C,
Mhm.
but I think C is not correct.
Why you think C is not correct?
Mm, I don't know about helicase well.
Okay, guys. Um, Tomohiro, what about you? What have you chosen?
I chose D.
Mm-hmm. And why?
In previous question, we learned that DNA polymerase is responsible in DNA replication, so I chose D.
Okay. Are you familiar with this word, um, unwinding, guys?
Make the structure of the helix into straight, right?
Yes, so let me show you the picture. Okay. Here, on this part, we see the unwinding of the DNA double strands. Okay. So here, the enzyme which is responsible for unwinding, so basically uh, breaking the bonds between the nitrogenous bases and basically pull apart one strand from the other. This is the unwinding. And in the previous question, I was talking about that in general, one type of enzyme are responsible for unwinding. Okay. Um. So Tomohiro, you went for right? DNA polymerase. Uh, Hitomi, you went for C. Mina, what about you?
I chose B.
B. Okay.
Yeah.
Okay. And why?
I guess.
Okay, you guessed. Mm-hmm. Okay. Let's discuss first of all why none of you went for A. Where or when is RNA polymerase involved? Do you know?
RNA polymerase is responsible for the process of transcription.
Perfect. Great, Hitomi. Thank you very much. So RNA polymerase is involved during transcription. Perfect. What about DNA ligase? Mina said that she went for DNA ligase, but it was more a guess. What about you, Hitomi and Tomohiro? Do you know about, do you know something about DNA ligase?
I remember that DNA regards connects the pieces of DNA chain, and so
Mm-hmm.
maybe it is not.
Okay, we're getting very close. So I will show you the picture. If you remember, please zoom in the picture and I will use this as a template. If you remember the process of replication, we mentioned that during DNA replication, we have two not similar strands. One is leading strand. We call it leading strand because on this strand, DNA is continuously synthesized. However, on the other side, we have a lagging strand. That's because that's because this strand is synthesized from 5' to 3', as you see. And if we have a 5' to 3', it's not there is no problem for DNA polymerase to synthesize DNA. However, the other lagging strand is in the direction from 3 to 5. And unfortunately, DNA polymerase is not able to synthesize DNA in new direction. So that's why That's why we create those primers here, basically a starting points from where DNA starts the synthesization of new strand. And as a result of this fragmentally or partially creating a new strand, we have Okazaki fragments. Okazaki fragments are named after one very famous and very important Japanese molecular biologist who found that. And uh, basically, those Okazaki fragments need to be removed in order to have absolutely complete double-stranded two double-stranded DNA molecules. And if we want to remove those Okazaki fragments, we need, let me show you, let me show you, we need DNA ligase. Okay, so DNA ligase plays a role in later stages of DNA replication and it seals the gaps between the newly synthesized DNA fragments. So it seals Okazaki fragments on lagging strand. Okay? So even though DNA ligase is involved in the DNA replication, it's not involved in unwinding. Okay? What about DNA polymerase? When is DNA polymerase involved? DNA polymerase, if we get back again to our picture, we see the DNA polymerase is just the enzyme which synthesize new complementary DNA strand. Okay, so DNA polymerase creates a new DNA polymer, okay, or it synthesizes something, as well as RNA polymerase in transcription creates new strand of mRNA. DNA polymerase also creates a new strand in the during the replication. So that brings us to the last option, that the unwinding process, which is necessary for creating enough space and breaking the bonds between the nitrogenous bases, belongs to helicase. Okay, helicase. Helicase is the right option if we are asking which enzyme is responsible for unwinding the DNA double helix during the process of DNA replication. In general, now I will repeat myself, in general, helicases are enzymes which do unwinding, okay? Unwinding. Guys, clear so far? Yes? Can we move on? Very well. So, um, Mina, please, can you read?
During which phase of the cell cycle does the cell prepare for nuclear division and ensure that DNA is accurately replicated? A. G1 gap 1 phase. B. S synthesize phase. C. G2 gap 2 phase. D. Mitotic phase.
Okay, so here are the results. Let's discuss them.
Sorry, I want to change my answer.
Okay, so
I
Hitomi,
chose B, but
yeah.
the she's I think she's correct.
Okay, so we will discuss.
Sorry, I also want to change my answer.
Mm-hmm.
I chose C, but maybe I think that A is correct.
Okay, so um, Tomohiro thinks A is correct. Hitomi, you think C is correct. Mina, what about you?
I chose B.
B. Okay, so all of the
Yeah.
options are correct. Uh, were taken. Only D not, except D. Okay, and why, Mina? Let's start with you. You were reading, so why you think B is correct?
Uh, Interphase prepare for cell division. So interphase include gap 1 and gap 2 and synthesize phase.
Mm-hmm.
Also, so synthesize phase, I think replication is chromatin. I don't know.
Yeah, so during S phase, DNA
Yes,
rep
yes, yeah.
during the S phase, DNA replication takes place or is happening. Okay, yes, that's right. So you would go for B. The question is, during which phase of the cell cycle does the cell prepare for nuclear division and ensure the DNA is accurately replicated? So, again, it will be better if we see a diagram. You very nicely explained, Mina, that the interface is a phase or big phase with three subphases which prepares the cell for cell division or mitosis. We divide interface into three other subphases, G1, S, and G2. During G during S phase, the replication replication takes place in order to have enough genetic material in both daughter cells. We mentioned that during G1 we have a checkpoint, first checkpoint, when the cell checks whether everything is okay and whether everything is properly functioning inside the cell. The other checkpoint was here, also in G2 phase, when just before the mitosis, mitosis, cell one more time controls, checks, or supervise, or whatever we will use, whatever word we will use, and and see if everything after G1 DNA replication and also the last uh, preparations after the last preparations before mitosis was well handled. Okay, so we see that we have two checkpoints. One is before DNA replication, one is after DNA replication. If we get back and we see during which phase of the cell cycle does the cell prepare for nuclear division and ensure that the DNA is accurately replicated, which option do you think now is correct if you have to choose one more, if you could choose one more time? Okay, guys, here are the results. I want to ask, am I using maybe confusing or not understandable words? Because you went for A or B, and the correct answer is actually C, G2. Um, I think I will try to explain it one more time, okay? During which phase of the cell cycle does the cell prepare for nuclear division and ensure that DNA is accurately replicated? So the question asks, in which phase the DNA is already replicated, so the replication was already done, and check or ensure that the replication was handled well, and also the other preparations are still going on before the mitosis. Okay, so this is the question. And if we see the diagram and we know that replication is happening during S phase, only one phase after replication is G2. So, and in G2, cell still prepares itself for mitosis and also check or ensure that the replication was handled well. Okay, so during G2 checkpoint, we are already done with DNA replication. DNA replication was already uh, processed during S phase. Okay, so if I ask you one more time, next, like maybe tomorrow, will you know that the G2 phase or during G2 phase there is a checkpoint when the cell check and ensure that DNA is replicated well and prepares itself for mitosis? Is it understandable? Or do you have any questions?
Yes.
Perfect. Tomohiro, also for you because you were not nodding.
No, no question for me.
Okay, so basically, if you will, if all of you guys just do yes, I believe you that you understand. Okay, if no, please ask whatever kind of question what is unclear. If I'm when I'm using maybe words you don't understand still yet, or maybe it's not in explained in comprehensible way, we will try to repeat and recap, yeah, in order that everything will be clear what we covered. Okay, so don't hesitate to ask questions, and hopefully now it's clear why C is correct. Okay, perfect. Let's move further. Tomohiro, please, can you read?
During which phase of mitosis do the sister chromatids separate and move towards the opposite poles of the cell? A. Prophase, B. Metaphase, C. Anaphase, D. Telophase.
Mhm. one more person. Okay, okay. So, Tomohiro, what have you chosen and why?
I chose C, and anaphase is the phase which contains the division of uh, no separation of chromatids, and they move to the to to opposite parts.
Mhm. Okay, perfect, exactly. So let very quickly, as I see that mitosis is already a topic you are familiar with. During prophase, the chromosome are condensation of chromosomes going on, and also creation of mitotic spindle, and disintegration or disappearing of nuclear membrane is also going on. And during metaphase, we see double chromatids chromosomes are aligned in the equatorial plane, and as well exactly all of you went for correct option, that during anaphase we see the pulling uh, apart or away of sister chromatids to the opposite poles of the cell. So basically a separation of sister chromatids going on in anaphase. And during telophase, chromosomes are decondensed, and there is a creation of new nuclear membrane, and cell division is taking place on the equatorial plane. And then the also cytokinesis, let's say, could be described as an individual phase. So basically, when the cell division takes place on the equatorial plane, we can say that it's already a cytokinesis phase. Okay, perfect. Hitomi, can you read?
During which phase of meiosis does crossing over occur? A. Phase 1. B. Metaphase 1. C. Anaphase 1. D. Telophase 1.
Thank you very much, Hitomi. Okay, guys, so C is the option. You went all four. Okay. Let me show you the meiosis one more time. Guys, if I will show you this chart or this picture, where do you think or where do you see crossing over is going on?
Metaphase to anaphase.
The or what is crossing over? Do you remember what the crossing over is from yesterday?
Chromosomes exchange their genetic information each other.
Yes, perfect, exactly. So we were drawing something like this basically. If you remember that we have two chromosomes, then the crossing over happens, and the outcome is that they exchange the some parts of their chromosomes in order to increase variety and diversity in the offspring. Here we see it, crossing over. Crossing over takes place during prophase 1. Okay? During metaphase during metaphase again the chromosomes are aligned on the equatorial plane, and during anaphase the homologous chromosomes are separated from each other, however with both chromatids. Okay, so not just sister chromatids are torn away, the whole chromosomes are torn away, pull apart, separated to different poles. Okay, guys, please remember that if there is one thing you should know about the meiosis is that it is it's this. When crossing over takes place, crossing over takes place during prophase 1. So during meiosis 1 and during its basically first phase, which is prophase. Okay, please try to remember that because um, it's really very important and very probable that the question like this will occur also in your admission questions. Okay, mitosis, meiosis, they are very often those topics are very often the part of admission questions, and it will be very unfortunate to lose points on these things which we are already describing and repeating many times. I know that it can be confusing, but please try to memorize that crossing over takes place during prophase 1. Okay? So do you understand that why metaphase, anaphase, and telophase are not correct? Because crossing over takes place basically during prophase 1, not during the other phases. Okay?
Yes.
Perfect. Um, so Mina, please, can you read?
What is a key difference between mitosis and mitosis in terms of the number of cell division and the number of delta cells produced? A. Mitosis involved two cell divisions and produce four daughter cells, while mitosis involves one cell division and produce two daughter cells. B. Mitosis involved one cell division and produced two daughter cells, while the mitosis involved two
Meiosis.
cell division.
Meo
Meiosis,
Meiosis.
mitosis involved two cell divisions and produced four daughter cells. C. Mitosis involved two cell divisions and produced two daughter cells, while meiosis involved one cell division and produced four daughter cells. D. Mitosis involved one cell division and produced four daughter cells, while meiosis involved two cell divisions and produced two daughter cells.
Thank you, Mina. I know it was exhausting, but thank you very much. A lot of reading. Great. Let's have a few more seconds, guys, and then we will do a poll. One more person. Okay, perfect. So all of you went for B. Great. Guys, you are absolutely right. Mitosis involves one cell division and produces two daughter cells, which are absolutely identical, while meiosis involves two cell divisions, meiosis 1 and meiosis 2, and produces four daughter cells, which are not identical, which are different, and all of them are haploid. Okay? So mitosis produces two daughter cells identical diploid daughter cells. Meiosis 1 and meiosis 2 are two cell divisions, and together it produces four daughter haploid cells. Very well. Perfect, guys. I'm really proud now on you. Um, okay, Tomohiro, can you read?
Which term describes the observable physical or biochemical characteristic of an organism resulting from the interaction between its genes and the environment? A. Genotype. B. Phenotype. C. Array. D. Homologous.
Thank you. Okay, perfect. All of you went for B as well, guys. So I really glad that you were listening and very good listeners yesterday. Phenotype. Um, Tomohiro, what does the genotype refers to? How to understand genotype?
Genotype is determined by the combination of genes only.
Yes, all combinations of genes, but basically, uh, the genotype possess or is referring to all genes within the organism. Yeah? So all of the genes, all the genetic material basically uh, uh, re, uh, is referring to genotype. Very good. Yesterday we draw that equation that genotype plus environment equals the phenotype. What about allele? Hitomi, do you remember what allele is? Hitomi, do you hear me?
Um,
Yeah.
yes. Mm, I can't explain. Sorry.
Okay. Anyone else? What about what the allele is? Allele is basically the form or variant of a specific gene. We mentioned that um, there are different alleles, or in other words, different variants of genes responsible for the color of a eye. If your eyes are blue and mine are brown, we have different alleles of the same gene which encodes color of the eye. So allele is just a variant of one specific gene. Understandable now, guys? Perfect. And what about the last uh, term, homozygous?
Two same areas.
Sorry,
No.
Tomohiro, can you repeat? I didn't hear properly.
Uh, it's a type. The combination of genes. It contains two same allele.
Yes, perfect, exactly. So, um, so many things in our bodies are encoded via more genes, and we have basically homologous chromosomes. One is from mother, one is from father. And if the gene here or the allele of the gene which encodes, for example, a blood group is the same on the father's chromosomes, in the same allele as on the mother's chromosome, we are homozygous regarding this trait because our allele are the same. Okay, so homozygous is an individual has two identical alleles for a particular trait. Okay, so two identical forms of a gene. We were describing yesterday that we have something like we draw it like AA or AA or no or small lowercase AA. Guys, if A if A is an allele, and here we have two alleles. Here we have two alleles, and here we have two alleles. And homozygous mean that homozygote is an individual with two identical alleles. Which of those three are homozygous?
large A,
Large A and also small a. Okay,
small A.
because those alleles are also identical, also same. Okay, those are identical, those are identical. These in the middle, um, lower and higher case, A's are heterozygote. Hetero, okay? Homo always means something from Latin what refers to something similar, okay? Homo. Hetero refers to something different. As you see, we have here different letters. One is uh, big letter, one is lower letter. So they are not the same. They are this is a heterozygous type. Okay, let's have two more questions. Who reads now? Hitomi, you?
Yes.
Okay, go on.
Which term describes a genetic condition in which an individual has two identical alleles for a particular gene, either both dominant or both recessive. A. Phenotype. B. Genotype. C. Allele. D.
Homozygous. Thank you.
Homozygous.
Perfect. I'm glad that that you were listening. What I was trying to explain to you, and hopefully it was well explained because all of you went for correct option, homozygous. So if we are describing genetic condition in which an individual has two identical alleles for a particular gene, either dominant or recessive, both, we are talking about homozygous. Great. Phenotype we discussed in previous, genotype as well, allele as well. Let's do the last one. Mina, can you read?
What is a basic unit of heredity that carries genetic information and is passed from parents to offspring? A. Alleles. B. Chromosome. C. Gene. D. Genome.
Yeah,
Genome.
C we can also read like gene.
Gene.
And A is
Gene.
allele, B chromosome, and D genome. Mhm. Okay, very good. Thank you very much, Mina. Let's do it. Okay, so here are the results. Let's discuss them. Mina, what have you chosen and why?
I chose C. But so I think so. Genes and genomes. So I don't know the difference
hmm
from
Okay, okay.
gene.
Um, the correct option is C, and we have to, I guess, then differentiate difference between allele, B, chromosome, gene, and genome. So first of all, how are genes, or how we can understand gene? Gene is a basic unit of heredity that carries genetic information and is passed from parents to offspring during reproduction. So it's a hereditary or basic hereditary unit, a gene. Okay. Also, genes are the segments of DNA located on chromosomes. Okay, so basically they contain some instructions for building and functioning of various molecules, proteins, and also they are very essential for the functioning of the cell, overall development and functioning of an organism. So basically genes encodes always some kind of information which are then very essential for the cells and for the whole organisms. So this is basically a gene, a segment of DNA located on chromosome. Okay, chromosomes on the other hand is a chromosome is a structure in the cell that contains DNA and a lot of genes. Okay, so chromosome um, contain DNA and a lot of genes, and chromosomes are composed of many genes arranged in linear order, as we saw, for example, during metaphase. Okay, during metaphase, it's very nice to see chromosomes aligned on the equatorial plane. Let me just show you very quickly where we are. Where is our here? Here, okay. This is structure of chromosomes aligned on the equatorial plane, or on this picture, chromosomes are metaphase. Metaphase is basically here. They are aligned. Okay, this is an anaphase already. Um, so where we are? Yeah, chromosome. Then gene we explained basically that is just a segment of DNA and a basic unit of heredity, and it carries some information. And last, genome. Genome refers to the entire set of genetic material. So genes and also non-coding, no-codings regions, we call them introns. We will get there today, which are present in an organism. So it includes all the genes found on the chromosomes of one individual or species. Okay, so genome is all of the genes and chromosomes together inside our bodies, inside the organism. Chromosome is a structure that contains DNA and genes, and gene is just a basic unit which is located on chromosome, and it's specifically or it encodes specific information for producing proteins or other essential various processes in the cell. Understandable so far, guys?
Yes.
Perfect. Okay. Um, would you like to have a break? Yes, no? Okay, so all of you want a break. Let's meet then in 16:11 here, okay? Hello everyone, welcome back. Okay guys, so we will continue where we ended yesterday. Okay? Um, Please, can you just remind me which terms we have covered and which we are still missing, or where we ended? Do you remember, Hitomi, Tomohiro?
Uh, we take group 1
Yes,
homogenesis.
so we did till homozygous, right?
Yes.
And on in group 2.
In group 2 heterogenesis.
Okay, so we only started with heterozygous. Okay, perfect. Because in the other group we ended on this slide, but in with different terms, so I wasn't sure. Thank you very much, Hitomi. Okay, guys, so yesterday we started with this, and it was also your group work, so we will continue with that. Hitomi, you were part, you were group 2, or basically was responsible for the list belonging to group 2. Can you tell us what are your findings regarding pure line?
Uh, and pure line is a population of plants or animals all having a particular feature that has been retained unchanged through many generations.
Perfect. Yes, exactly. So, um, pure line is basically a set of organisms crossed in a way to have the same traits in each generation, a set of homozygotes. Basically, exactly as you said, it's nowadays mostly belonging to plants and animals. However, the pure line was already known long before the dawn of the genetics. And exactly, so basically the pure line or the generation have the same traits as you said. Perfect. What about dominant expression?
And dom- sorry, I could only look up to the fourth word. Dominant expression and recessive expression is opposite word. And dominant expression is tend to appear as a genetic property.
Okay, so I can maybe provide you with the understanding or definition that dominant expression of an allele or phenotype manifests whenever it is present. So what does it mean basically? If we have two alleles like this and then two heterozygotes in this way, and let's say that the outcome would be the color of a plant, the this individual will have a blue appealing or blue leaves. Okay, so I will draw a plant with blue leaves, and dominant expression or dominant allele will, in this case, even though we have different alleles, have the same phenotype or manifestations because dominant allele masks the present of an recessive allele. So only in this case would be leaves in a green color. Okay, guys, this is just an example. This is just an example. Okay, I'm just trying to explain what does it mean dominant and recessive expression. Recessive expression of an allele or phenotype appears only when present in both copies or in a recessive homozygote. So this is our case. regarding recessive expression. So what I want to emphasize, if if we are dealing with a dominant expression, that basically means that homozygotes as well as heterozygotes will have the same manifestation. Okay, so they will look the same, even though their genotype are different. Okay, understandable?
Yes.
Perfect, perfect. Um, Hitomi, you mentioned that you didn't complete the whole list. So what else have you found out? From which terms?
And recessive expression is hard to appear as a genetic property.
Yes, so basically what we said that a recessive expression will manifest only when when we have a recessive homozygote. What about complete dominance? Have you found something like this, Hitomi? No?
No,
Okay,
sorry.
okay, perfect. So you done the first four, right? Okay, so we will continue now. So complete and incomplete dominance, it can be more confusing now, but complete dominance in heterozygotes means that only the dominant trait is expressed. Complete dominance basically means what we just discussed. If we have a complete dominant in heterozygote, it means that only this allele, the dominant allele, is expressed. But if we have incomplete dominance in heterozygotes, there is a blend of both traits, blend or mix. So the outcome would be something between blue and green. So it will be like it will be appearing like this. Okay, some kind of mix. Both alleles plays a role in a manifestation, and then we will get to codominance, because codominance is something different than incomplete dominance. Codominance is not basically the thing, um, or how to say, when we say incomplete, when we are still dealing with incomplete dominance about this case, we can say that we are not far from the truth to say neither of these traits is dominant. They are more like being equivalent in this case. So one more time, I don't want to confuse you. So one more time, we will repeat what we know. Okay? If we have a homozygote with AA, with two dominant alleles, the appearance would be that in our case, we have a blue leaves on the plant. If we are dealing with a dominant expression, even though we have heterozygote, which alleles are different, the manifest would be still the blue because we have a dominant allele and dominant expression. And complete dominance is basically what describes the relationship between those two alleles. in homozygote or recessive homozygote or recessive expression, also dealing with the recessive expression means that recessive expression can be manifested only when we are dealing with homozygotes which have both recessive alleles. So only in this case we have a green leaves. However, in incomplete dominance means that I will try to draw it one more time. We have a big allele, big A letter, which wants basically which wants to have leaves in blue color, but then lowercase allele or recessive allele, which wants to have leaves in green color. But in this relationship, in incomplete dominance, they are more or less, let's say, equal. So the outcome is that the manifest of the leaves would be something between. So the blend basically, or a mix of those. Okay, so far,
Yes.
okay, perfect. And codominance, we will use a little bit different because codominance and incomplete dominance are many times uh, misunderstood. In codominance, basically both alleles are expressed. It's very hard to differentiate it in our case of leaves, so we will use something different. We will use our blood types group AB0. If you remember, we were discussing once red blood cells that we have group A, we have group B, we have group 0, and then AB. Okay, do you remember that? This uh, um, AB0 blood group system. So let's imagine, guys, now that our parents are, let's write, our father is blood group A. Our mother or your mother is blood group B. So the possibilities of the offspring and their blood group are basically all of them. We will get there, but for a more simplified way, we will discuss it in this simplified case. My possibility as an offspring of my parents is that I will have a blood group A, or I will be, as my mother, blood group B, or I will have a both from them and I will be group AB. Why is that possible? Because in a blood group system AB0, we have a codominance uh, relationship between the two alleles. So both alleles are expressed simultaneously, not just in a mixed version, that there we have one mix, something between green and blue, as we have in incomplete dominance, but both alleles A and B are expressed in their way. Okay, so if I would be an offspring with A and B group, even though my father is just A and my mother is just B, it's possible that I'll be AB, AB blood group, because there are possibilities, or there are cases, or in other words, in blood group types, there is a codominant, codominant, codominant relationship between genes. So each variant or allele of gene is expressed equally. Okay? Okay. Guys, so far so good. Do you have any questions?
No question.
Okay. Tomohiro, can we go back to your list regarding parental generation?
A parental generation is the previous generation of organisms.
Okay. Okay. The parental gen. How can you repeat? Can you repeat one more time?
Parental generation uh, is the previous generation of organisms.
Yes, previous. Sorry, I didn't hear this word previous very well. The parental generation is a starting point of a cross. So basically, usually consists of homozygotes for a specific trait. One dominant and the other recessive. Yes, it's a basically previous generation. In many theoretical cases, we are dealing with homozygotes in parental generation, but also heterozygotes could be there. But for simplifying for simplifying and for better understanding, we use mostly homozygotes. But basically, parental generations are in the cases parents, okay? And they are the triggering or the starting point of crossing. Then and we will get to the first filial generation. First filial generation is the first generation of offspring, typically set of heterozygotes. So if we are dealing with now some genetics genetics cases, first of all we write down parental generation and then first filial generation, which are their offspring. And that is also the very similar to second filial generation. So basically, second filial generation or F2 are offsprings of our offsprings. So F1 or filial F1 or filial generation becomes an parents and they have their own offspring. So we call it this way. Why what's going on here? Okay. Okay, so second filial, monohybrid and dihybrid. What does it mean? So far, we were dealing only about monohybrid cases. Monohybrid is a cross. How to say in other words? Maybe to be more clear. Uh, uh, I will try to find maybe crossbreed or hybrid. Do you understand, guys, that crossbreed or hybrid? Crossbreed. I will try to find Japanese translation. Fu.
雑種、
Zasu. Maybe I will try to put it here. Yes, do you understand?
Yes.
Perfect. So, if we are dealing with a monohybrid, it's a Zasu involving just a single trait. This is our monohybrid. Okay, so we are dealing just with a single trait. So far it was just a color, let's say, of our leaves. If we have a dihybrid, we have a crossbreed involving two traits. Okay, so we draw it like this: AA and BB. What does it mean? That another trait besides a color of the leaves could be the the length of the roots, for example. Okay, so the length of the root are determined by those B genes or B alleles. So far it is dihybrid. We are already involving in our dihybrid two traits. One is color of the leaves, and second one is the length of the roots. Okay, so monohybrid and dihybrid both are crossbreeds. However, when we are dealing with the genetic examples or cases, we are talking about some traits. And if we are looking for just one trait, we have a monohybrids. Okay, so involving only one trait. If we are discussing already two traits, we are dealing with the dihybrid. Mhm. And it would be same for trihybrid, but we would do just, you know, this, let's say AA, BB, CC, and another trait would be involved. So maybe the color of an or the height of a flower. Okay, how high the flower is, let's say. Okay, so the flowers are higher or smaller. This can be also encoded uh, via different allele or genes. Okay, let's get back. So leave it here. Let's get back to genome. We already discussed that. However, Tomohiro, can you provide us with your findings?
About genome,
Genome.
the genome is all genetic materials in an organism.
Yes, the set of all genes, yes, in the organisms. Okay. Exon and intron.
Uh, exon is the area of RNA which is remained in after splicing, and intron is the area of RNA which is thrown away after the splicing.
Perfect, exactly. So intron is a part of mRNA that is removed and not used after transcription. Because, guys, if we get back to the transcription, there is an, let's say, I will draw a DNA. Yes, this first part represents exon, and the black one will be, no, 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 no. I need to do it differently. One more time. DNA. This is exon, this is another exon, this is another exon. In the middle, we have introns. Okay, introns are only for reading. The scientists are still not sure what are the main purpose of introns. However, they play no role in protein synthesis. Okay, so if we now synthesize our mRNA, we also copied copied the exons but also the introns. So after mRNA there is some processes or steps after transcription when introns part are removed, and as an outcome, we have only exonic exonic part of mRNA. Okay, so introns are not used after transcription, and exons participates in the protein synthesis. Understandable, guys, so far? Okay, crossing over, Tomohiro, what have you found out?
Crossing over is the exchanging of genetic materials between chromosomes, which is occurring in prophase 1 or
Prophase 1, exactly. So crossing over is an event occurring in prophase 1 which increases tremendously the variability of an offspring. Okay. Let's get back to finish the group 2 list. So linkage group, it's a group of genes located on one chromosomes which may or may not undergo crossing over. So basically, if we have on one chromosome more genes next to each other, let's here we have more genes, we can call them, it's a linkage group. Okay. um, Gene mutation. Gene mutation is a change in one or more nucleotides in a gene. May or may not affect the resulting protein. Mutations include deletions, inversions, and substitutions. We will get there later. Let's see maybe just a picture for simplifying it. So here on this slide, we see some chromosomes changes, uh, basically mutations. Okay, so mutations are also part of evolution. And the last one, chromosomal mutation or aberration, a change or loss of an entire portion of chromosome. Mutations include numerical and structural mutations. Okay, so again, we will get there later. On this picture, we see the chromosomes break um, and structural mutations and changes. Okay. So let's move further to classical Mendelian genetics, guys. Do you have any questions to those terms?
Sorry, please explain group 1 intron and exon.
Intron and exon one more time.
Exon, please.
Yes, so I will explain it now with the definition first of all. Okay, so exon is a part of mRNA that participates in protein synthesis after transcription. One more time, exon is a part of mRNA that participates in protein synthesis after transcription. And intron is a part of mRNA that is removed and not used after transcription.
Thank you.
Okay, you're welcome, Hitomi. So, classical Mendelian genetics, guys. Um, okay, we will start it because we need to move on. Group work, or I think it will be now individual work. Mm, or no, we can do group work, why not? But you can start with the first Mendel's law together. Okay, you will start together. So it will be a collaboration. And then second and third group we will do together in the main room, okay? Because you are a very small number today, I think it won't be as useful for you to work individually to distribute the workload. However, I think you can deal with the first Mendel's law. So guys, I'm going to give you this picture or this slide. And now your task will be, okay, we have to end in 8 minutes. So you have 8 minutes to start with the research of first Mendel's law. What does it mean and how this slide, what does this represents? Because we already discussed many important facts or parts which are necessary for understanding the Mendel's first law in the previous describing activity. Okay, so this is basically your task. Try to find that out. You have 7 minutes, so in 7 minutes I will turn my camera back again, and so this will we will you will just start with the research, and tomorrow we will continue with this activity. Is it okay for you guys, or is it okay for you?
Yes. Ah,
Okay, perfect. So please try to discuss. I'll be here, but I do not want to step in when you are in the teamwork. Okay? So if you have any questions, ask it. However, please discuss. Okay, what are your findings? What have you just find out? So as usual, group work.
すいません。有用なサイトを見つけたんですけれども、
あ、
これルームチャットに送っちゃっていいもんですかね。
送ってほしい。
送って、
と、
ありがたい。
ありがと
これが
う。あ、
多分
本当だ。
わかりやすいと思います。
ありがとう。
ありがとう。
ほんまや。
あ、本当だ。
Okay, guys, how is it going? I know you are middle in of the process. Can we stop it here and we will continue tomorrow with Mendelian genetics? Yes?
Yes.
Okay, do you have any questions, guys, so far?
No question.
Okay, so thank you very much that you participated today, even in a small number. On the other hand, I think it's better because you have more space to discuss, to ask questions, and more space to for you. So thank you very much, and yes, see you tomorrow, guys. I'm looking forward to see you. Thank you very much that you are still attending the classes. That's great approach.
Thank you.
You're
Thank you.
Thank
welcome.
you.
See you. Bye.